The broadcast is now starting. All attendees are in listen-only mode.
Hi everybody, my name is Ed Davis. I'm Senior Applications Scientist at GeneCopoeia Incorporated, located in Rockville, Maryland, just outside of Washington, D.C. I want to thank you all for joining us for today's webinar entitled Unleash the Power of Luciferase to Analyze microRNA Activity in Cancer. We really appreciate it. Before I get into delivering the webinar, I just want to go over a few practical matters. First, you may ask me questions at any time. But in order to do so, you won't be able to speak verbally to me because your microphones will be muted. Instead, you'll need to type your questions in the questions box in the control panel. If you can't see your control panel, it might be hidden. So in that case, look for a small strip of buttons on one side of the screen. One of the buttons will be orange with a white arrow pointing in one direction. And when you click on that button, you'll then be able to see your control panel and hence the questions box. And next, I will, even though you can ask questions at any time, I will wait till the end of the webinar to actually answer your questions. And when I do so, I will read each question out loud for everyone to hear, as well as read out the answer for everyone to hear. And I will answer each question in the order in which it was received. And finally, I will send you a link at some point to download a PDF if you want to, and I will do that through the chat function, which is also located in your control panel. All right, so with that, I will get started. So first, I want to tell you who we are at GeneCopoeia and what we're about. We are a biotechnology company that focuses primarily on functional genomics, molecular biology, and cell biology. And we were founded back in the late 1990s as a company that provided a large, essentially genome-wide collection of human and mouse open reading frame or ORF clones, ORF plasmid clones. And these are mainly used for people doing cell culture work, but they could also be used for bacterial expression. But they're expression-ready clones that can be used the day you receive them. And since then, we've branched out into many other clone types, such as those for promoters, microRNAs, which of course you're about to hear about today, CRISPR genome editing, and shRNA. But in addition to that, we also have products for viral systems, mainly lentivirus and AAV, where we'll produce infectious viral particles for people. On top of that, we have a number of stable cell line products, such as CRISPR-Cas9 stable cell lines, labeled cancer cell lines, and cancer biomarker cell lines. Further, and you'll hear about some of this today, we have a number of kits and reagents for transfection, for luciferase, of course, that we're going to hear about today. Fish probes for fluorescent detection, indel detection, and cloning. And finally, we have a large collection of products for fluorescent detection, mainly like cell function assays for apoptosis and other cell functions, nucleic acid detection, cell structure probes, and fluorescent dyes. But of course, today you're here to talk about both microRNAs as well as luciferase. So let's get started. All right, so here's an outline of the seminar. First, I'm going to give you a basic background on microRNA biology, and then I'm going to give a brief introduction to luciferases. And then I'm going to talk about two examples of published papers where the customers or the researchers who are customers of GeneCopoeia used GeneCopoeia microRNA and luciferase products to analyze microRNA function in two different types of cancers. So again, there are two published examples that I'm going to discuss. And finally, I'm going to go over other ways of analyzing microRNAs. All right, so what are microRNAs? Well, I'm sure you know this. They're small non-protein coding RNAs. They're a class of such small non-protein coding RNAs that are about 21 nucleotides in length. The length does vary. It can be 20, 21, 22, even 23, but it's usually around 21. They're present in most known metazoans, including and especially humans and mice. There are approximately 1,900 known microRNAs that are known to exist in humans. There may be more, but that's about the number right now in the latest mirror base. And their main known function is to post-transcriptionally regulate levels of protein coding gene expression by targeting the 3' untranslated region or 3' UTR of messenger RNAs. And it does this either by destabilizing the messenger RNA for degradation, or through imperfect complementarity with the mRNA through inhibition of translation. But the bottom line is they cause a post-transcriptional downregulation of gene expression in most cases. So microRNAs go through a fairly complex biogenesis cycle. They start out as what has been termed miRNAs. These are like pre or microRNAs. And then these are processed in the microprocessor complex by an enzyme called drosha to form a pre-microRNA. Pre-microRNAs are then exported to the cytoplasm, where they're further processed by dicer to give rise to a mature microRNA, which is what interacts in the argonaute complex with messenger RNA for either, again, mRNA degradation or inhibition of translation. All right, so let's talk about a little bit about luciferase. So luciferases in general are a class of bioluminescent enzymes. These are like special chemiluminescent enzymes. So like a good example of a chemiluminescent enzyme is like alkaline phosphatase, which converts a substrate into a light signal. Well, luciferases do something similar, but they catalyze, they're enzymes that convert a substrate to light. Okay, and in this case, so the substrate is different for some luciferases. For example, the substrate for firefly luciferase is luciferin, and the substrate for Ranella luciferase as well as Gaussian luciferase is silanterazine. And luciferases are found in many organisms, mostly like beetles and sea creatures. Like a good example of beetles, again, is a firefly. I don't know if you knew that firefly was a beetle, but it is. And then, but also some various sea creatures like sea pansies, of which Ranella is a sea pansy, and copepods have a number of luciferases. And one of the most well-known copepod luciferases is from Gaussian princeps. But luciferase is also found in other organisms. And one of the key features of luciferase is that they have very high signal-to-noise ratios due to virtually complete absence of background in mammalian cells. So that's unlike GFP, which, even though GFP is widely used, there was even a Nobel Prize for the discovery of GFP and its use in biology. Fluorescent proteins like GFP tend to have some suffer from high background due to autofluorescence from the organism. But that's not the case with luciferase. There's essentially no background. Okay, so let's talk about the published examples I alluded to for studying microRNA in cancer. Okay, so microRNAs are very important players in cancer biology. They're obviously essential for normal cell function and development because they're so critical for gene regulation. And so, as you might expect, their dysregulation can be pathogenic, and they're found in many diseases. The dysregulation of microRNAs has been shown in many diseases, especially cancers. So microRNAs were first shown to be involved in cancer in a deletion occurring in chronic lymphocytic leukemia, or CLL, where loss of microRNA function leads to overexpression of BCL2, which is a known inhibitor of apoptosis. So they've since been found to be involved in pretty much every stage of many cancers, and their stable presence in body fluids make microRNAs a good predictor of prognosis. And so microRNAs, of course, are known to be heavily involved in cancer as both oncogenes and tumor suppressor genes. So microRNAs have been found. This table shows some of the many genes that microRNAs are regulated by. And as you can see, there's a very busy list, but there are some well-known genes that are involved in cancer, such as PTEN, BCL2, I mentioned before, CDK6, FOXP1, et cetera, et cetera. So some of these, again, because of their downstream regulation of genes, some of these microRNAs are either oncogenes because they downregulate tumor suppressors like PTEN, or they're tumor suppressor genes because conversely, microRNAs like LET7 downregulate oncogenes like NRF, NF2, etc. And so obviously there's been many examples that have been shown for microRNA involvement in cancer. So in the first example I was going to discuss, this is an example of a group that works on prostate cancer from Latonen et al. From earlier this year. So prostate cancer is the most common cancer among men in Western countries and the second most common cancer among men worldwide. And there's no cure for castration-resistant prostate cancer, which is a further developed form of prostate cancer, where even by castration, the cancer is still resistant and comes back, and it's very deadly. It's found compared with untreated primary prostate cancer. This group found in this paper, compared with untreated primary prostate cancer, or PC, they found that there were 728 proteins differentially expressed in benign prostate hyperplasia, which is non-cancerous or precancer, and they also found 382 proteins that are differentially regulated in castrate-resistant prostate cancer compared with untreated primary prostate cancer. And they found that there were 153 proteins that were common between the two sets. They went on further to show that 95 microRNAs were differentially expressed between CRPC and PC. And pathway analysis indicated that the tricarboxylic acid cycle was strongly differentially expressed in CRPC versus PC. So when they noticed this, they found a strong signature for MDH2, which is from the enzyme that encodes malate dehydrogenase. So they found that MDH2 protein levels are differentially expressed in CRPC versus PC, while the microRNA levels themselves, the messenger RNA levels themselves are not. And so in light of that, they found that two microRNAs, miR22 and miR205, are differentially expressed and negatively correlate with MDH2 protein levels. And so what they ended up doing is they expanded further by testing the effect of miR22 and miR205 on MDH2 mRNA expression. And to do this, they used a GeneCopoeia microRNA 3' UTR clone as an assay system. So here's an example of a generic microRNA clone from GeneCopoeia, and the idea of these clones is that they contain the 3' UTR of a messenger RNA, which is the target region for a microRNA, and that is linked to a reporter gene. Okay, so in this case, what this group used is the Gaussian Luciferase, which again, as I mentioned, is an enzyme from the copepod Gaussian princeps. And the nice thing about Gaussian Luciferase is that it's secreted. Secretion allows you to assay the luciferase activity in the medium instead of breaking up the cells like you have to do with Firefly and Renoa luciferase. So in these clones that the customer used, the Gaussian luciferase is driven by a constitutive promoter, but it's linked to a microRNA target 3' UTR. Okay, so the idea is if you express this in cells in which the microRNA is absent, you will see a downregulation of Gaussian luciferase activity. And also on the same plasmid is an enzyme known as secreted alkaline phosphatase, which is also a chemiluminescent enzyme and that's not regulated by a microRNA. And so this is constitutively expressed. And SCAP is here on the plasmid as a transfection loading control. So again, the idea is, with GeneCopoeia, we clone the 3' UTRs from any human, mouse, or rat gene fused to this luciferase reporter. Again, microRNA activity will cause a decrease in reporter signal. And it's a dual reporter vector system, as I mentioned, and these are ready for transfection in mammalian cells. So the customer in this paper, the Latonen et al. Paper, ordered a 3' UTR clone containing the MDH2 3' UTR fused transcriptionally to gosylluciferase. And so when you transfect these clones into cells, again, they will cause expression of the gosylluciferase as well as secreted alkaline phosphatase. These enzymes are secreted into the medium, and then you assay the medium. Okay, and again, the secreted reporter system is great for live cell assays. Lysis is unnecessary. And this is a sensitive and robust detection system, so even though firefly luciferase and vanilla luciferase are widely used, gosylluciferase is actually 1,000 times more sensitive than firefly and vanilla luciferase. And in the GeneCopoeia gosylluciferase assay kits, we provide 2 robust buffer conditions that can be used either for promoting stability or high signal strength. An example of that is shown here. These are using promoter clones, but it's the same idea, where the gosylluciferase gives very robust signal compared with negative control clones. This is using a GAPDH clone, or different promoters like EF1 alpha, GAPDH, and MYC. And then the panel on the right shows the 2 different buffer conditions I mentioned. So if you use buffer GLH in the GeneCopoeia secrete pair assay kit, you get very high signal strength, but the signal decays rapidly over time. So these are good for short-term assays. But if you use the GLS buffer, that's for promoting stability. And what you see is, even though it's a lower signal, you see a very stable signal over time. And so the Latonen et al. Paper used both the GeneCopoeia my target 3' UTR clone as well as the GeneCopoeia secrete pair luciferase kit to do this assay. And so they co-transfected PC3 cells, which is a prostate cancer cell line, with microRNA plasmids and the GeneCopoeia my target 3' UTR clone. And so they used the miR22 and miR205 microRNA plasmids. And so what they observed was a small but statistically significant reduction in luciferase using the GeneCopoeia secrete pair kit. And so these results suggest that microRNA activities might play a major role in prostate cancer progression. Now, this is not necessarily a causative result. It could be a correlation, but it's still at least a suggestive result. Okay, so now I want to talk about the second example for studying microRNAs in cancer, second published example. And this is from Moore et al. Back in 2017, where they're studying acute myeloid leukemia. This is from a paper published in Cancer Cell in 2017. Acute myeloid leukemia is an aggressive blood cancer, and it begins in the bone marrow, and it's characterized by enhanced proliferation, blocked differentiation, and dysregulated apoptosis. And it's known that Hox genes which are involved in normal development, Hox genes play a major role in AML development and progression. So what they found, one of the things they found is that both HOXA9 and MICE1, MEIS1, which are protein coding genes, when overexpressed in lineage-derived bone marrow cells, transformed those cells, and then when they transplanted those into immune-compromised mice, they killed the mice. So they made very aggressive tumors. Proteomics analysis showed that 1,810 proteins are differentially expressed after this transformation event. And SYK protein is one of the most upregulated proteins in these HOXA9 mice1 transformed cells. However, as in the previous example, SYK mRNA is not upregulated by using either QPCR or RNA-seq as an assay. And they found that, they did find that SYK protein, but not mRNA, is overexpressed in patient AML samples. Further, they found that SYK protein overexpression in the absence of microRNA elevation suggests that microRNAs might be involved. And so they did global microRNA expression profiling using RNA-seq and found that 8 microRNAs are significantly downregulated in cells overexpressing HOXA9 in mice 1. And one of these 8 microRNAs, miR146A, is predicted to bind the SYK 3' UTR at 2 different sites. And so that brings us to our next microRNA target clone product. I mentioned before that we have that our microRNA target clones, GeneCopoeia microRNA target clones, are microRNA 3' UTRs, not microRNA 3' UTRs, protein coding 3' UTRs, fused to reporter. In the previous example, it was fused to Gaussian luciferase, but we also have clones that are fused to where the microRNA target is fused to firefly luciferase. Okay, but the principle is the same. If microRNA activity is present, you will then get a downregulation of the firefly luciferase signal. And also in these plasmids, we express luciferase constitutively in order to have a good loading control. So again, just like with the Gaussian clones, these clones are 3' UTRs from any human, mouse, or rat gene. And then, of course, microRNA activity will cause a decrease in the reporter signal. It's a dual reporter vector system, so the renilla luciferase serves as a transfection control. These are ready for transfection in mammalian cells. And best of all, these are compatible with any firefly or renilla luciferase assay system. Now, GeneCopoeia, of course, does sell firefly and renilla luciferase assay kits under the brand Luciferases, but you can use them with other companies' firefly and renilla luciferase assay kits. So here's the key data from the second paper I was mentioning from Moore et al. So what they did was they co-transfected HEC293 T cells with plasmids expressing the microRNA that I mentioned before, as well as a GeneCopoeia MyTarget SYK 3' UTR clone. And again, just like similar to the first paper, they observed a small but statistically significant reduction in firefly luciferase activity, which was normalized to renilla luciferase activity. And these results suggest that microRNA activities might play a major role in AML formation and or progression. So a similar type of result to the previous paper, but the main difference is that the previous paper used the Gaussian luciferase system with secreted alkaline phosphatase, and this paper used the firefly and renilla luciferase system. However, we always strongly recommend using the Gaussian luciferase and secreted alkaline phosphatase system because, again, the secreted proteins allow the great benefit of doing live cell analysis. So now I just want to briefly talk about other ways of analyzing microRNAs. You can measure microRNA abundance by qPCR, and to do that, you can use the GeneCopoeia all-in-one microRNA qPCR primers. These are primers that are designed using a proprietary algorithm. They're pre-validated, so when you order them, we do make them to order usually, although we do have many that have been made before. But we will only ship them if they actually function in a qPCR assay. So you get the added benefit and peace of mind of having a pre-validated primer set. And these work best when used in combination with GeneCopoeia's all-in-one CyberGreen microRNA qPCR kit. The features of which are shown here. These are designed for, before I talk about the kits, I also wanted to talk about microRNA qPCR arrays. We have high throughput profiling arrays are designed for high throughput profiling of microRNA expression. And again, the primers are designed using a proprietary algorithm, and they're pre-validated. And we have 43 pre-made arrays that are available, including whole miRNA, of which we have 5. We have 27 different cancer-related microRNA qPCR arrays, including for like breast cancer and prostate cancer, etc., as well as disease. And we have 11 disease and focus group microRNA qPCR arrays, which are not cancer focused. And in addition to our pre-made cancer microRNAs arrays, you can also have custom microRNA qPCR arrays made, pretty much anything you need. And they're best when used in combination with the all-in-one CyberGreen microRNA qPCR kit. We have a number of different reagent solutions for microRNA qPCR reagents. This is a table of the different kit options we have. I mentioned we have a microRNA qPCR detection kit. It comes in 2 different sizes. One is a 200 qPCR reactions and 600 qPCR reactions. And these also contain reverse transcriptase, so you can just isolate your RNA from samples, do reverse transcription to get cDNA, and then use that with our all-in-one qPCR polymerase to run a qPCR assay. Or you can also buy the the first-strand cDNA synthesis kits separately. So you have a number of different options. In addition to the qPCR arrays and reagents, as well as the 3' UTR target clones, GeneCopoeia also sells microRNA precursor clones. And so what these do is these express the precursor microRNAs, which, as I mentioned earlier, are processed by Drosha and Dicer into mature microRNAs. And these come in 3 different plasmid backbones that we have shown here. The difference between these is mainly the reporter gene, and we also have non-viral and lentiviral options. But these are microRNAs that are fused to fluorescent reporters, and they're driven by constitutive promoters. And so when you express these in cells, they are then processed to the mature microRNA form. And so these are ideal for validating microRNA binding sites in 3' UTR clones, which is like what was done in the 2 papers that I mentioned. These clones also are full coverage. We have all known human, mouse, and rat microRNAs in the current MIR-based covered. They're available in flexible delivery systems in either lentiviral or non-viral format. They're also available as ready-to-use lentiviral particles. And but we also, if you don't want to use plasmids, we also have synthetic mimics available. And then conversely, to go along with the microRNA precursor clones, we also have microRNA inhibitor clones. And so what these do is these plasmids express single-stranded microRNA, antisense microRNA that are designed to competitively inhibit microRNA function. And these are available in 5 different vector types shown here. Mainly, again, the differences are due to either the presence of the fluorescent reporter, and again, they're also ideal for validating the binding sites in the 3' UTRs. So, but in this case, you're doing a competitive inhibition with the microRNA precursor. There's also full coverage for these clones. They're available also in lentiviral and non-viral formats, or as lentiviral particles, or you can purchase synthetic RNA. And then finally, I did talk briefly earlier about the secrete Paragosi luciferase assay system, but we also sell, and I mentioned this before, Firefly and Vanilla luciferase assay kits under the brand name LukePair. There are seven different types of kits shown here, and they're mainly used for different purposes, such as the our top seller, the Leukemia Duo Luciferase Assay Kit 2.0, assays both Firefly and Manila luciferases, and this is designed for enhanced stability. Okay, so it gives a very stable signal. But in addition, we also have a high sensitivity assay kit. So usually, you when you enhance stability, you sacrifice activity, and the opposite is true. If you sacrifice sensitivity, if you enhance sensitivity, you sacrifice stability. So the HS assay kit is designed also as a dual luciferase kit, and it's designed for high signal intensity. We have a similar kit called the Dual Luciferase HT Assay Kit. That's for high throughput, and it comes in ready to use buffer solutions. So they don't need to be diluted, and it's ideal for use with injector systems. And so those are the Dual Luciferase Assay Kits. But we also have Leukemia kits with either Firefly or Luciferase only, for either enhanced signal intensity or high throughput, for stability, or high throughput, as well as a Ranilla only kit. And just to give an example, GeneCopoeia's luciferase, Firefly and Ranilla luciferase assay kits perform very well compared with our competitors. This is a comparison of the Leukemia 2.0 kit compared with a competitor dual Luke system. And as you can see, both the Firefly and Ranilla luciferases give comparable stability between the two kits. Okay, so if you want to know more about GeneCopoeia's luciferase system, you can download a technical note that we have available. This tells you all about how to choose a GeneCopoeia luciferase system, whether it's for Gaussian luciferase or Firefly and Ranilla. This is the download link, but I don't expect you to memorize this or to write it down. Instead, I'm going to copy and paste it into your chat box. And so you can look for that, and if you want, you can download it at any time from now to the end of the webinar. So just look for that in your chat box. Okay, so with that, I would like to summarize. I told you that microRNAs are important post-transcriptional regulators of gene expression, and their dysregulation can lead to many types of diseases, including cancers. I told you about the GeneCopoeia 3' UTR plasmid clones, which have been used extensively to help demonstrate the critical nature of microRNA function in the formation and progression of cancers. And I must emphasize that I did talk about 2 papers, but there are many others. I just picked out those 2 as good examples. I also told you that luciferases are bioluminescent enzymes with high signal intensity and low background, which makes them ideal reporters for detecting microRNA activity in cancer progression. And finally, that GeneCopoeia offers many products from clones for 3' UTRs, microRNA precursors and inhibitors, to powerful secreted and Firefly Ranilla luciferase assay kits to greatly facilitate your cancer microRNA studies. All right, so with that, I'm going to stop here and take questions. Okay, so the first question is, can I get the slides? Yes, you can. After the webinar, I will make both a recording available for on-demand viewing, as well as the slides available for download. So yes, you can get the slides. Okay, next question. Does GeneCopoeia have adenoviral vector for 3' UTR? Well, the answer is for adenovirus, we actually don't carry any adenoviral products. We could make them. We could make an adenoviral 3' UTR clone, but we don't normally have that. We don't even have the 3' UTR clones available as luciferase clones for various reasons. Now, if you mean adeno-associated virus, we we could possibly do that. We do have adeno-associated viral particle options. So you would just have to contact us for a custom project. Okay, next question. Can I use your microRNA QPCR arrays with the Applied Biosystems PCR machine? We do have the arrays are custom, usually custom built to comply with the specifications of a large number of qPCR machines, especially including many Applied Biosystems machines. So if you have an Applied Biosystems machine, it's highly likely that our arrays can be used in your machine. Yeah. Okay, next question. Does GeneCopoeia's qPCR enzyme work with FFPE samples? Yeah, that's actually a good question. So they should. The answer is they should. You just need to isolate your RNA using methods that are optimal for FFPE samples. Because I'm sure, as you know, FFP RNA samples tend to be degraded. Even though microRNAs are small, you still have to be careful. But yeah, it should be no problem. Okay, next question. What filter do I have to use for luciferase? We get that question from time to time. Now, so luciferases do have emissions wavelengths. Like, for example, Firefly is, I think, like 550 or 560, and Renilla is 480. But it turns out you actually don't need to use filters for luciferases, especially when you're using a luminometer. And the reason is that there's no excitation input, light input. And so it's not like fluorescent protein like GFP, where you shine light on it and then it's excited at one wavelength and then it emits at a different wavelength. With luciferases, there's no background and there's no excitation, and so you get light output, but it's the only light that's detected in the cell. So I hope that makes sense. But yeah, you shouldn't have to use a filter for luciferase. All right, so those are good questions. Are there any more questions? Okay, well, if not, then I would like to, again, thank you for joining us for today's webinar. And if you have any further questions, please give us a call, 1-866-360-9531, and my extension is 227. Or you can email me, edavis@genecopoeia.com. And please visit our website to learn about the many different products that we have for molecular biology at www.genecopoeia.com. Thanks, and have a great day.